Hi, I'm Judy Cole, the Executive Vice President and CEO of the MIT Alumni Association, and I'm delighted to welcome you to this web production of the MIT Alumni Association.
Welcome to the MIT Faculty Forum, Alumni Edition. I am Ann Gibbons, a Knight Science Journalism Fellow from 1987-88 and a correspondent with Science Magazine. This is an interactive forum, so we encourage your questions. Use the Google form below the live stream to ask a question or tweet one to hashtag #MITBetterWorld. We'll try to get to as many questions as possible. Today's topic, will immunotherapy save us? It's been called a game changer in the field of cancer research and healthcare by either stimulating or suppressing the patient's own immune system in various ways. Researchers have discovered some radical new approaches to combat conditions as wide-ranging as the common wart to lung cancer, which is estimated to kill more than 150,000 Americans this year. So we're going to start with each panelist. I will ask each one to introduce himself or herself and give us an overview of their current research or immunotherapy. So we're going to start with Krishna. He's going to be our first. Krishna Komanduri, class of '87, is a professor of medicine at the University of Miami Miller School of Medicine. Krishna, you can go ahead if you're ready.
Okay. Thank you, Ann. About your work. Do I need to do anything to activate the video?
We see you, so maybe you just go ahead.
Okay, great.
If not, we can,
Perfect.
Yeah.
So I'm Krishna Komanduri. I'm as Ann noted, thank you, Ann, professor of microbiology, medicine microbiology and immunology at the Sylvester Comprehensive Cancer Center at the University of Miami. And I wear several hats. The first is I direct the hematopoietic stem cell transplant program here at Miami. And as many of you will know, blood and marrow stem cell transplantation is really now a 60-year-old field, but is really one of the first forms of we know, successful cellular immunotherapy. So when we do blood and marrow stem cell transplants, we are transferring both stem cells from donors, but also immune cells that we know to be critical for the success of hematopoietic transplantation. By my laboratory work, I do study T cells, their development and their function. And for a long time, our laboratory has been interested first in studying how healthy T cells, CD4 and CD8 T cells, which are subsets of T cells that have different functions, recognize viruses that are normally in our environment and can cause disease, especially in immunocompromised people. So by understanding how our immune system responds to pathogenic viruses, we've learned a lot over the last 2 decades about how T cells develop and how they get dysregulated in the context of other diseases, including immunodeficiency diseases following chemotherapy and in the setting of cancers. So our work over the years has evolved to trying to better understand immunologic consequences in the area where I'm most clinically interested, which is in the area of cancer and hematopoietic stem cell transplantation. So again, when we transfer a donor immune system into a recipient, a lot of things can happen. First, the donor T cells can help to protect the recipient against pathogenic viruses, but the donor T cells can also cause disease via target organ damage in recipients, but they also mediate graft responses that are really essential for the control of malignancies in these donor transplants. We now, over the last decades, really, I think in part due to the early success of again, allogeneic or donor transplants as an immunotherapeutic modality, recognize now that immune system cells, independent of chemotherapy, can have dramatic effects in cancer. And I think a lot of our conversations today, and I'm really excited to hear what other panelists think, will be in areas where we've seen dramatic evolution of cancer therapies. And these include checkpoint inhibitor therapies, where we know that if we take the brakes off immune regulation, we can get activation of the immune response against cancers where cancers are not normally susceptible to immune responses. And also in the last few years, and with a lot of movement in the last 2 years in particular, we've seen the rise of gene therapies involving T cells that recognize specific antigens. And these include what we call CAR T cell therapies, or chimeric antigen receptor T cell therapies. Our clinical site at the University of Miami is involved in a number of these CAR T cell and adoptive T cell transfer studies, including by KITE and Juno and Adaptimmune. And I will say finally that I wear another hat. I'm the president-elect of the American Society for Blood and Marrow Transplantation, which is a professional society for many of us who are in the field of hematopoietic cell therapies, and are in that way, I'm involved with a lot of ongoing behind-the-scenes discussions about how the future of immunotherapy will be regulated with respect to how cells are produced and how the financial and local regulation of these therapies will end up impacting, I think, their ultimate clinical use. So that's a summary of my background, and I'm looking forward to this discussion. Thank you.
Thank you very much. Marcela Maus is the Director of Cellular Immunotherapy at the MGH Cancer Center in Boston. Marcela, greetings, and we'd like to get a quick summary of your work and what you think is exciting in immunotherapy.
Sure. Thanks, Anne. Can you hear me okay?
Yes.
Wonderful. So actually kind of following up a little bit on what Krishna was talking about, my work is also really focused on turning T cells into cancer fighters. So we know how to genetically modify and engineer T cells now to be able to target specific antigens on tumor cells. And T cells are these fantastic cells because they have these two somewhat unique properties. One is that they can kill other cells, and the other is that they have this very long life, and they have what's called memory. So they can go back and recognize an antigen they've seen before, a target they've seen before, and kill it very efficiently, to the point where somebody doesn't get sick again from the same virus. This is why we're immune to measles and chickenpox after we see it once. And so we've now discovered ways of engineering T cells to redirect them to specific cancer antigens. So I've been involved in both making novel kinds of CAR T cells. CARs are chimeric antigen receptors. It's a way that we use to redirect tumors, to redirect T cells to specific tumors. So I've been involved in making new forms of CAR T cells, translating that into patients by designing and implementing phase 1 clinical trials. So first in human studies with these cellular products, and then running the trials associated with them. And some of the work we do in the lab is to generate these novel forms of T cells. We're engineering new receptors into them. We are trying to change where T cells go and what they see and how they respond to the tumor microenvironment. And we're also trying to figure out ways to make the whole process a little bit more manageable. Right now, it's a pretty complex logistical effort to try to get the right cells engineered in the right way back to the right patient. We still don't know which patients are most likely to respond and which ones will not. And so we're trying to make this a little bit more scalable and easier. And I also, like Krishna, I'm involved in some of the regulatory framework. This is a completely paradigm-shifting new kind of drug. There's no other drug that is like a cell. The closest to it is bone marrow transplant, which is why people like societies like the ASBMT and the Foundation for Accreditation of Cell Therapies are involved. But those have never been for sale before. So the combination of using a cell as a drug is really a novel paradigm for our healthcare system. And so really understanding how that's going to work both in the regulatory framework, the financial framework, and how these cells are going to be administered clinically, and how they work on a scientific basis is all sort of going to be sorted out, I think, in the next couple of years. And I'm interested both in the basic science of making new cells, the translational aspects of turning that into products that can be used in humans, and then sort of larger scale of how we're going to turn those into cancer fighters that are available for any patient.
So that's great, right on the front lines there. Zoltan Maliga, let's go to you next. He's a research associate at the Harvard Medical School.
Hi, thank you very much, Anne. My name is Zoltan Maliga. I'm a research scientist at the Laboratory of Systems Pharmacology at Harvard Medical School. I basically wear two hats. One of them is actually drug discovery. I'm doing small molecule discovery to find new ways of activating macrophages. These are cells that exist in the tumor microenvironment that actually downregulate the immune response against cancers. And it's a new therapeutic modality. The idea is that by this way, instead of having the immune checkpoint inhibitors now that are currently in vogue, this might be another generation, a new way of turning on the anti-tumor response. My second hat is actually with the Ludwig Cancer Center and the LSP doing very interesting histological measurements in tumors. And a question there is there's a lot of different types of, whether you're looking at conventional cancer chemotherapy or immune therapy, the the tumor environment is actually a very heterogeneous place with many different types of cells. And the question is, can you look at tissues themselves to figure out what potential therapies are best targeted, and also what patient populations will respond to certain therapies with minimal side effects? So one aspect that's actually quite interesting is many of these immunotherapies, they are fantastic when they work, but they can also have extremely toxic immune side effects that must also be managed. And getting on that early is good for sort of patient compliance and getting better. Thank you.
Thank you. Yes, we should come back to the toxicity issue and how immunotherapy can be used to actually sometimes turn off aspects of the immune system. And for that, Jonathan Maltzman works in that area. He's an associate professor at Stanford University School of Medicine in Palo Alto, and he's interested in transplantation and how to, I guess, downregulate the immune system. So Jonathan, your turn.
Thank you, Anne. Do I get video or no?
We see you.
You can see me, good.
See you.
As Anne said, I'm a physician scientist at Stanford University and at the Palo Alto VA healthcare system. And I was an undergrad at MIT, graduated in '89. And right now what I do is I run a laboratory that's interested in T-cell biology that's related to transplantation, but also I do see patients occasionally that are kidney transplant patients. And what I would say is that my side of this is now for something completely different. And so what I did want to do is put up one slide for you to see. And let's see if I can do this, which is really something that I think about all the time, which is the balance. So hopefully you're seeing that slide now.
Yes, we can see it.
So I think about the immune system as a balance. And what you've heard from the other three speakers is that they're trying to change the balance so that they can have more of these effector type of cells that would fight off the cancer. I think about it from the other side, which is trying to increase the regulation or increase the ability or decrease the ability of the immune system to reject or fight off a transplant, which is the major problem we have in solid organ transplantation. And let's see if I can go back to me. Did I turn off the screen share?
You did.
Good. Basically, keeping a solid organ transplant, be it kidney, liver, heart, or lung, relies on continuous immunosuppression or immunoregulation. And there's really been very little change in terms of the drugs that we're using clinically over the last 15 years. The one change has really been a drug that targets co-stimulation, and it's exactly the opposite of some of the cancer immunotherapy checkpoint inhibitors that are being used now, and I think that that'll come up as we discuss things. So that's a drug called belatacept. In my laboratory itself, we really study two things. One is one arm of the immune system which increases regulation, which are called cells called regulatory T cells, and the more of those you have, the less immune function overall that we think you're going to have. And this is a potential source of cellular therapy. And the other thing that we study is how the immune system continues to respond to chronic viral infections in the setting of transplantation and immunosuppression. As Marcela mentioned, you have these memory type of T cells, and we have to be able to make them last and continue to fight off things like cytomegalovirus, while at the same time making sure that they don't reject a transplant. And I'll leave it there.
That's really interesting, and we want to come back to that, the promise with transplantation, since tolerating immune suppression, tolerating not having a huge immune response is critical to the success of those transplants. So we're going to start with questions now, and I'm going to start first with Tasha's question, Tasha in Wisconsin. She asks about side effects, and she asks, it seems like immunotherapy has less side effects and better results than chemo. Are there reasons why chemo would still be considered a better alternative? What else needs to be done to make this treatment available to more patients? So that's two questions. Let's unpack it. Let's talk about, are there less side effects with stimulating immunotherapy than regular chemo? And how do you see these two therapies working together? And let's start with Marcela, and then I think we can see where to go from there. Can you hear me, Marcela?
Yeah, I can. I just had trouble
Good.
unmuting for a second. Sorry. So, it is an interesting question. Does immunotherapy have fewer side effects? I'm not sure that I would characterize it that way, but I think the side effects associated with immune therapy are often related to the function of immune therapy. So, for example, when we give CAR T cells, we sometimes see that patients no longer have a different kind of immune cell called the B cell when we're targeting their lymphoma or their leukemia. But that's an expected targeted effect. It's not the same thing as losing your hair to try to get rid of your breast cancer. They sort of, in this case, losing your B cells goes hand in hand with the way the immune therapy works. So I wouldn't characterize it necessarily as having fewer side effects or safer side effects. They're different. What I do think is appealing about them is that it's not completely unrelated. It's not, as I mentioned before. In some cases, some of the immune side effects we don't completely understand yet. When we give checkpoint antibodies, particularly in solid tumors, there can be rashes associated with it, or other target toxicity, other organ toxicities that we don't fully understand how they're related to the cancer yet, because activating the immune system is still kind of a broad, using a very broad drug, and so there is a chance that there can be significant side effects with it. So I wouldn't say that it's completely less than chemotherapy. There are different side effects, and we need to understand those a little bit better. There are times when chemotherapy, I think, is still very much warranted, and it's not going away anytime soon. In lymphomas, for example, somewhere around 80% of patients can be cured of some kinds of lymphoma with chemotherapy alone, and it can be an intense period of time, but once it's over, it's over, and I think that can be worthwhile. There are also drugs that are on the shelf that we know how to use very well as clinicians, that we know how to manage their side effects, and so I think in some cases it's just very useful to have a very rapid onset control of disease, and then know that we haven't really changed the long-term spectrum of what the immune system sees and recognizes. So I think there's going to be a role for chemotherapy. That said, I'm still pretty excited about immune therapy, not just scientifically, but because it has this potential to be curative in areas where chemotherapy is not. So I think, both have a role, and it remains to be seen exactly which patients should get which form of therapy. And that's going to become, the balance may shift over the course of the next few years, but that's still always going to be a patient-physician conversation.
So that's Krishna, who has seen some side effects that are pretty serious with CAR T cell therapies. They're exciting and promising, but sometimes I guess the question of when you can turn them off, it can be an issue when they maybe overkill. So Krishna, can you address that issue?
Absolutely. So I would just highlight the fact that most of the approaches that are currently being utilized with CAR T cells actually still do administer chemotherapy first to recipients, and that chemotherapy typically knocks down the amount of cancer that's there, as well as helps to clear space for the immune system and allows the infused T cells to expand. So the first thing I would note is that most of the approaches that we expect to be seen approved with CAR T cells are not completely independent of chemotherapy. I think all of us hope that there will be, at some point, a selective immunotherapy that does not depend on chemotherapy or other combinations, but at that time, that's not the case. I think it's important to note right now that we have seen some pretty significant toxicities, arising in CAR T cell therapy studies over the last several months. And in fact, one of the, I think, important contributors in this space, Juno Therapeutics, which was founded really in the basis of intellectual property from Memorial Sloan Kettering Cancer Center and the Fred Hutchinson Cancer Research Center, which has reported really some dramatic results in highly refractory cases of acute leukemia. That the Juno trial, called the Rocket study, was halted by the FDA earlier this summer, and then last week there were 2 additional deaths in patients on that study that were very unfortunate, that led the company to actually place a voluntary hold pending further FDA review, and also the review of their data monitoring committee, which was reviewing safety for that trial. So there is a very, I think, intense, appropriately controlled regulatory environment around these studies. And I think, as really what we have seen in the context of these studies, is that there can be both expected toxicities. We know that when these chimeric antigen receptor T cells expand, there can be an inflammation, which is associated with the appropriate expansion of those cells. But in the context of the recent studies of what we call these CD19 CAR T cells, CD19 is a target on both healthy and malignant B cells, including those in subsets of lymphoma and leukemia, that there have been unexpected neurological toxicities. It appears that those neurological toxicities happen more in the acute lymphoid leukemia patients than in others. And indeed, many of the deaths in the Juno study were due to swelling of the brain that really remains a bit unexplained, because we don't know, for example, that there is CD19 expressed within the brain. So I do think that I would agree with Marcela that we can't just simply say that immunotherapy is somehow non-toxic. Chemotherapy can certainly be well tolerated under some circumstances. So we're going to see evolution, and ideally what we'll see is the evolution of safer therapies in this tight regulatory environment.
In fact, Leo from Naples asked that some have criticized the FDA for not holding up clinical trials of immune-stimulating drugs after patient deaths. But what you're telling us, it sounds like these drugs have been held up. And the flip side of that is, what is the research that's being done to work on those kind of side effects? And I think Zoltan can answer that. Well, maybe not. Maybe you can't answer it, but you can talk about the kinds of research trying to address those issues with
Yes, thank you. So at this point, the antibodies that are essentially targeting the immune checkpoint, these immune checkpoint inhibitors are effectively becoming more frontline therapies. And one of the reasons that is there's been a significant learning curve over the past few years to realize that a small subset of the patient population, I mean, it can be curative if the immune toxicity can be managed. Some of these are quite severe, skin peeling off, really severe GI symptoms as well. But over time, people have done, through rapid blood testing and histological measurements as well, people basically titrate in immune suppressive drugs that will, or either antibodies or small molecules that are basically suppress the immune toxicity to allow the anti-tumor effect to occur, because the patient can tolerate the treatment. I think that's really the main point right now, is that as these immune therapies become a bit more mature and there's a large armamentarium of the drugs that we can choose from to treat the cancer. So for instance, the CTLA-4 antibodies originally were extremely toxic. I think now with the PD-1 inhibitors, these are now becoming a better drug of choice, and combination therapies themselves are becoming even more effective. But here it's just key to suppress some of the T cell responses that can be toxic in certain people. Like the big issue is looking for biomarkers, either biomarkers for response and also biomarkers for toxicity. I think that's.
And so we had a question also from that's related to this from Jessica in New Jersey, where she says, will genetically modifying T cells have any negative effects on the body? Are there any potential issues with these newly designed T cells attacking healthy tissues? And I thought that might, I'm not sure which one of you is best for that, but Jonathan Maltzman might be interested in that with transplant issues.
So it's a very interesting question. I think that when you genetically modify a T cell, you're really targeting it against something. And it's really what everybody else has said. If you're targeting a molecule called CD19, it's going to target all of the B cells because they express that molecule. At the same time, if there's someplace else that expresses that molecule, you're going to target that as well. So another example might be if you were to try to target melanoma with a CAR T cell, you'd be targeting a protein that's specific to melanoma. The problem is, or could be, that if normal cells also express that protein, you're going to target those cells as well. And therefore, you would have some kind of autoimmune consequence.
So I want to move to some of the more promising, we're talking a lot about the side effects, but let's talk about the really promising diseases that might be possible to treat and where they are in the pipeline of, trials and drugs at this point, and the possibilities of when patients will see them. Related to that, we have a question from Carol in Atlanta. She says, it seems to me like manipulating immune system with new kinds of cells would have dramatic impacts on allergies. And I'm wondering if any of you want to take a stab at that. Have any of you thought about allergies, or then if not that, what other diseases are the low-hanging fruit that might be some of the first ones we will treat? And I'm thinking either Marcela or Krishna, perhaps.
I can comment briefly. I'm sorry that I think allergies are actually very difficult. There have been some therapies aimed at IgE, which is, again, an antibody that's important in many allergic conditions. There have been also approaches to try to desensitize patients, giving, ironically, low doses of these antigens, which can help to induce tolerant states. But I think that there's actually been less work. Ironically, I think that even though the market and the potential number of individuals that affected is extraordinarily broad, there has been probably more commercial development of pathways to try to activate the immune system against cancer because of the life-threatening implications. So, but I do think that, as Jonathan and I both know, approaches in general to deal with tolerance and better understand how normal cells like regulatory T cells interact with effector cells that mediate allergy, and in those cases, those can be both B cells and T cells. We may, again, be able to better understand those negative regulators in the future, but unfortunately, less progress than I'm aware of in those areas in recent years.
Marcela, do you have any thoughts on this question? What are it sounds like allergies are going to be a little more complex, and that's probably related to downregulating the immune system. What are some of the diseases that are on the horizon as the most, some of the first that we might be able to address with this therapy?
Yeah, so there's a lot of things that go into those decisions. Actually, I'm a long-term allergy sufferer myself, and that's why I got interested in immunology in the first place. But when we're talking about engineering new T cells, it's considered a high-risk therapy. I mean, there's just it's a very novel strategy. It involves genetically modifying someone's cells, ex vivo manipulation. And so there has to be a balance of risk-benefit. And right now, I think the closest balance is in cancer, and particularly in refractory leukemias or refractory solid tumors. So when the alternative is popping a Zyrtec or something, it's just really hard to justify going in with a novel therapy. And trust me, I know popping a Zyrtec doesn't answer everything, but it's just that the bar to accept a potentially life-threatening new or novel medication is just much higher in something like allergy. So I think, probably cancer is going to be the first indication for novel therapy, novel cell-based therapies. I think in terms of other forms of immune therapy, oftentimes I think we'll see that we'll start off in cancer. There's already several FDA approvals and for checkpoint therapies in solid tumors. And I think the next thing will probably be, bone marrow transplant, solid organ transplant, and then maybe really serious autoimmune disease if we can manage to understand scientifically how we can control regulatory T cells and immune suppressive cells. I think it's a lot easier to make a T cell kill than it is to make a T cell suppress, but there's a lot of science around making regulatory T cells and understanding really how these are different different epigenetic pathways, different signaling pathways than conventional T cells. And so if we learn how to manipulate those cells, then I think that there's going to be potential as well in serious autoimmune diseases like multiple sclerosis, lupus, etc., where there's serious disease.
Zoltan, did you want to add in to this? Where do you see these therapies being most likely to be applied first? But also, I did have a question from Ralph Lynn in San Diego, wondering if this has been overhyped, especially the CAR T, given the problems there. So I'd love a read from maybe you, Zoltan, on how close are we to actually having these therapies?
Well, I think that I'm less well positioned to be discussing the CAR T therapies themselves as opposed to immune checkpoint. So I'll talk about what I know a little bit better. So I think that the immune therapies are not overhyped insofar as they're in advance. So every time, in the very beginning, it was surgery, chemotoxic chemotherapy, then the magic bullets of targeted therapies. Now it's immune suppression. So effectively what we're doing is we're adding new tools in our ability to treat cancer. And what we do when you have every new therapy effectively meets some new unmet need. And that's actually very important because all these require clinical trials and patients suffering a lot. So effectively the motivation for that, the low-hanging fruit is to meet these clinical unmet needs. So I think in that regard, cancer immunotherapy is absolutely in advance right now because more people are living. I was married recently, and the person who was the event organizer in Canada, her husband was one of the first people enrolled in the Merck clinical trials for PD-1 agonists in Montreal, he had a life expectancy of 3 months, and he's been alive for 3 years now. And he's a long-term survivor, and that's probably curative. So I think that's actually a very spectacular result. That being said, I think that there's another, at this point, what might be good is, instead of looking at these macrophage T-cell interaction inhibitors, there may be ways of directly stimulating specific subsets of macrophages in tumor microenvironments that will sort of not even require these therapeutic antibodies, but have orally available agents. I think that's sort of the direction that some people are trying to move.
So I have a question. Would these therapies be something that you would go through an intensive period like chemo and then you'd stop the therapy, or is it something that would have to be continuous? And especially because it sounds like you're starting out first with patients who are not responding to traditional chemotherapy with leukemia or lymphomas and things like that.
Well, that's an excellent question. I think that one question is, do we have better patient stratification? So I think if we have a tumor biopsy, if we look at biomarkers, we may be able to know, should we give a person conventional chemotherapy or immunotherapy, which is now becoming the first-line therapeutic. And as these drugs advance, you're just going to have more tools that are available. I think it's really important now what we're doing with the histological approaches we're pioneering at Harvard Medical School, where you can, say, profile in the same tissue 20, 40 markers. You can start looking at what a patient tissue looks like in a clinical trial before, during, and after chemotherapy to get a better response profile for, what is a responsive patient, and how can we determine ahead of time which one of these therapies will be most effective to treat them.
Jonathan, a question for you. Often transplant patients end up having the rest of their lives are going to be on very difficult immune suppressant drugs. Do you see immunotherapy being easier to tolerate if it works out?
So that's an interesting question. It's something that people doing transplantation have been sort of grappling with for more than 50 years now. And there are a few different approaches. I'd say that everything that we do is actually immunotherapy. It's just a question of whether you think about it as cellular therapy, biologic therapy, or small molecule therapy. As I said earlier, we actually are starting to use biologic therapy that's exactly the opposite of the CTLA-4 blockade that's used in cancer immunotherapy. And that happens to give the opposite effect, which is immunoregulation or immunosuppression. That's something that needs to be used lifelong at this point. There is an approach to using, I'd say that there are two sort of approaches that I know of that both involve cellular therapy to induce more lifelong tolerance to a given transplant. Those trials are all going on in kidney transplant more than others. And one of them is to use regulatory T cells, which has been brought up by a number of people. So these are the cells that suppress the immune system. And the idea would be, if you can give enough, you can induce a change and a modulation in the immune system that might be lifelong. I'm not sure that that's going to work that way. The other way that's been used is combining tissue transplant with bone marrow transplant. And there are a few centers around the country. One of them is at Mass General, where Marcela is, where if you give bone marrow in a specific way along with a kidney transplant, you can wean other immunosuppression over time, and eventually those people can be off of immunosuppression for the rest of their life. And the places that those really going on is both here at Stanford, Mass General, and a third different protocol at Northwestern that I know about. So I think that that's potential cell therapy that could induce a way of not needing lifelong immunosuppression.
So I have two questions that are related to this and the future and how to proceed with this. And one is, doesn't the insurance industry support or respond to these new research and these new trials? And also that comes from Jacob in Amherst. And then we have a question also from Kevin in Weston, Mass, asking if international labs, especially in Europe or China, with looser restriction, are they ahead of the U.S. in trials? So these are both questions addressed at sort of, what are the barriers to having this happen? Let's start with you, Krishna, and maybe Marcela on that one, and we're getting close to our time limit.
Sure. I'd like to comment. So the first thing is about insurance. So if you ask insurance companies, and actually we had a pair forum around hematopoietic transplantation a few weeks ago, and I directly asked a number of these insurance individuals, executives, if you ask them publicly right now, they will comment kind of the way Apple doesn't talk about advanced products, that these are experimental therapies, and that they will develop approaches that will become public once they are approved. But I can tell you that behind the scenes, a lot of discussions are happening around payment models. I think what we see around hematopoietic transplantation, which is of course now decades old accepted therapy, will likely apply. That there will be probably a cost around the cell infusion, but there will also be a cost around taking care of these patients, which involves the treatment of toxicities, which in some cases will be minimal, but in some cases might be extraordinarily expensive. It involved ICU stays or even, unfortunately, or fatal toxicities. So the concept is a little bit different than giving a drug like a statin or other things in cardiovascular disease, where there really is not a variable hospital-associated cost with that. So it's likely that the payer framework for this will be around what we call the bundled payment system, where we pay for the drug, but also pay for care around the time of the cell infusions. It's similar to what those of us in the solid organ transplant and hematopoietic stem cell transplant communities are used to accepting as a financial framework. But the reality is right now, these trials are being paid for really by individual single payers, and those are the pharma companies, the biotech companies that are developing the therapies. So right now, if you need one of these therapies, you'll typically call up an academic center, and again, it could be an individual center-funded trial or a pharma-sponsored trial, but typically all of the costs that are experimental around that therapy will be borne by the person who is developing the drug, with standard care costs being taken care of by your insurance. So that's the current framework. The second question was about what's happening internationally, and actually, at the ASH meeting this year, I just saw an abstract. There's a hospital system, Lude Daopei Hospital System, which is a large academic cancer hospital system in Beijing, that's going to report the results of 47 patients treated with CAR T-cell therapies. I was at a meeting around post-transplant hematopoietic transplant relapse a few weeks ago in Hamburg, and there was a group in Hangzhou who reported over 10 patients who they had treated who had had relapses following autologous cell transplants for lymphoma, and reported encouraging results. I would say that the results that are reported are actually extraordinarily encouraging, with lower toxicity rates than I've seen from US studies. So I think that there will need to be some vetting and crosstalk and understanding whether they're really doing things, I think, in a novel and important ways where we can learn from, or are there limitations or caveats to those studies that we haven't seen yet. So I do think that this is something that's happening in a large number of sites in the US with a large number of companies, but clearly there are international sites, especially in China, that are making meaningful progress.
Thank you. That's great. And Marcela, I was going to go to you with sort of the same question. What is on the horizon? We've had questions in from people asking about boosting the immune system. For example, someone with with bladder cancer who, we have a question here from someone who has non-invasive bladder cancer, and his doctors gave him BCG, a regimen based on infusion of a form of bovine tuberculosis bacterium. I'm just wondering what is percolating and what you see developing and coming for patients sooner.
So, you know what, it's kind of an overwhelming question because there is so much now. I mean, immunotherapy is really undergoing this massive explosion of new science and new clinical trials, not only with new agents, but also combinations of agents. So, you know, it's too complicated to go into any one particular case, but there's lots of different forms of immunotherapy. I mean, there's antibodies, there's things like BCG, which stimulate immune responses, you know, in a localized area in this case, because it's infused into the bladder. There's cells, there's drugs that affect immune cells. So there's various, I think Jonathan Saltzman was talking about earlier, there's a lot of immune drugs that we already know affect the immune system and can control immune activation or less activation. So there's just, I think it's wide open. I would certainly say that there's tremendous excitement in the field. I think one of the challenges is actually going to be figuring out what are the best combinations and what can we really learn from them. Because, you know, statistically, if you do 100 studies and you say your p-value is significant at less than 0.05, then 5% of those studies will be meaningless just on a statistical basis. And so it's really going to be a challenge to kind of sort out where the signal is, and if there's a lot of noise in the background. But that said, one of the things that I like most about immune therapy, and particularly cell therapies, is that they are so powerful that sometimes the signal is just incredible. And this is what's happened really in the CAR T cell field. We were talking about whether there was hype or not. And, in some of these patients, it's 90% complete remission rates in patients who had progressed after multiple prior regimens. The disease was really refractory. And so I think whether it's going to work like that in all patients, we don't know. Whether it's going to apply to other cancers, we don't know. But it's just such a powerful system that if we learn how to manage the toxicity, and we learn how to figure out how to apply this to other cancers, it could really be revolutionary.
We have 2 minutes left, and I'm going to end with Zoltan. We have a question asking from Christopher in Washington whether the major advances are happening in industrial labs or university research labs. I don't think it's a competition, but it seems like industry is invested in a big way, right, Zoltan? You have
I think industry is invested in a very big way, but there's actually a lot of partnership between industry and academia. And I think you also have to realize that because of all the innovation that's happening in academia, there's a lot of spin-offs where essentially a 2 or 3 year old project that develops some IP will actually start and make a small biotech venture that can then grow either into a larger company itself through growth and venture funding, or else actually partnering up with a lot of pharma companies. So in my field itself, I know there's one company where you have a macrophage activator reagent that was developed in a company in California that's now partnered with Novartis for clinical trials, because that seems to be a potential therapeutic modality. So I'd say that a combination of private funding, a lot of business development also from major pharma as well, because they're looking for the innovation that's taking place both in smaller biotech and academia as well. So I'd say it's a very complex ecosystem that's going to launch the therapeutics forward. And of course, the large pharma is the only one that can really provide the money for developing real immunotherapy research.
very exciting. This is really good. I really appreciate all of you. Let me give a wrap up here. Panelists, I'm sorry, I have to find my end comments here. So on behalf of the Alumni Association, thank you for turning to this Faculty Forum Online, Alumni Edition. Thanks again to our alumni panelists from Harvard, MGH, Stanford, and the University of Miami. We didn't get to all the questions submitted, but we got to a lot of them. But rest assured, our panelists will see them all. Tweet about today's chat using the hashtag MIT Better World and send follow-up questions or feedback to alumnilearn@mit.edu. Thanks again for watching.
Thank you so much, Anne.
Thanks again for joining us. For more information on future MIT Alumni Association productions, please visit our website.